Okay everyone, back in the bus please. Hi, I'm your host Michelle Cordons, the True Fruit Specialist at Perennia. This is bus number 1. We've got 2 buses going, so just make sure that you got back into the right one. We've got a great lineup for today. We are going to be talking about bees, field crops, and also Christmas trees. And these are all under the topic of optimizing production. So it's going to be a great couple of stops. So hopefully you grabbed your coffee because the tour is about to begin. Okay, we're just pulling into our first stop where we're going to find Andrew and Monica on the topic of bees.
Bees and blueberries in Atlantic Canada. Wild blueberry is a major crop in eastern Canada that depends on insect pollination. The majority of this pollination is carried out by bees. Due to this, in Atlantic Canada, the working relationship between bees and blueberries and beekeepers and blueberry growers has been ongoing for many years. Every year, thousands of honeybee colonies are rented from beekeepers by blueberry growers and moved into blueberry fields to carry out the task of pollinating blueberry flowers in order to maximize the fruit crop yield. Honeybees are not the only bee species employed for blueberry pollination. In some fields, bumblebee colonies and alfalfa leafcutter bees are also often moved in to increase the pollination workforce. Bumblebees are effective in blueberry pollination due to their ability to perform a specific pollen foraging technique called buzz pollination, where they shake the blueberry flower, causing a greater release of pollen than other bee species are capable of. Alfalfa leafcutter bees are well adapted to our climate and are better able to forage at relatively low temperatures compared to honeybees. Honeybees are effective in blueberry pollination thanks to their populous colonies and generalist style of foraging abilities, meaning they are able to forage from a wide variety of floral sources instead of specializing on a small proportion of plant species. Given the low population of native pollinators and the high demand for pollination services on wild blueberry crops, honeybees are a common and important resource. Regionally, the current demand for pollination exceeds 50,000 colonies, and this is expected to increase. This represents over $8 million per year in income to Atlantic Canada's beekeepers. Although we have seen an increase in the number of beekeepers across the Atlantic provinces, the number of beekeepers supplying hive for pollination services has reduced. Increasing both the number of beekeepers providing pollination services and the number of colonies managed for this purpose has become a priority of both beekeeping and fruit production industries. When beekeepers and blueberry growers agree to use honeybee colonies for wild blueberry pollination, the use of a contract is recommended and should include specific details on the number of colonies required, the timing of placement and removal, who is responsible for providing and setting up protective fencing around the hives, the agreed on hive strength standard, hive inspection entitlement and arrangements, the location and access for delivery of hives, and any details concerning agrochemical use. As honeybees are managed pollinators, beekeepers and blueberry producers can be coordinated so that pollination services occur in optimal conditions for both plant and pollinator success. This coordination is supported by following a hive strength standard that sets a minimum requirement for the strength of honeybee colonies being used in pollination. It is important to note that the use of a hive standard for hive strength is not only beneficial in the case of wild blueberry pollination, but also in cases of other crops where honeybee colonies are employed to carry out pollination services. Hive Strength Standard. A hive strength standard sets guidelines for the minimum strength requirements for colonies being used for pollination, where colony strength is a measure of the population size and also the population structure of the colony. In other words, is there a queen that is actively laying? Is there sufficient numbers of foraging workers for resource collection and house bees to support brood rearing? All measures that impact how successful a hive would be in contributing to the pollination of a blueberry or other fruit crop field. Often, many hives are brought to a blueberry field together to perform the task of pollination, so the hive strength standard is considered as an average among all hives involved, with some colonies being above and some perhaps slightly below, but ultimately evening out to achieve the hive strength standard for pollination. The determination of hive strength and a hive standard is an often discussed topic in relation to the pollination of wild blueberries. There are acceptable standards recommended by provincial governments and national organizations, some of which are specific to the crop being pollinated, and some that are more of a general standard for pollination. A review of available hive standards demonstrates a range in the measures and criteria used to establish this benchmark. For the purpose of this video, we will discuss hive standards set by the following 4 organizations. First, we have the Canadian Association of Professional Apiculturists, or ATTTA. Then we will look at the Nova Scotia Beekeepers Association, or NSBA, hive standard, followed by the New Brunswick Department of Agriculture, or NBDA, hive standard, and then finally, the hive standard set by the University of Maine. The measures used to develop a hive standard are listed across the top of the table and include frames of bees, amount of brood, number of hive bodies, number of bees, and under additional comments, we can see that some standards consider presence of a laying queen and number of honey frames. Not all of these measures are included in each hive strength standard, but it is important to understand the reasoning behind each of these requirement factors. Frames of bees is an important consideration as this indicates the population of worker bees available in the colony to perform pollination services. The amount of brood is measured since it indicates the productiveness of the queen and how well the colony will be able to sustain the population of foraging or pollinating worker bees. The number of hive bodies is important to consider since this determines how much space the colony has to support brood rearing and resource storage, both of which are key factors in maintaining colony strength. The number of bees is a consideration to include and similar to the number of frames of bees, as it indicates the population of worker bees available to contribute to the pollination workforce. Some hive strength standards explicitly state that a laying queen must be present, as this ensures that brood production will continue throughout the pollination time frame. Some hive strength standards also require a certain number of honey frames to be present in hives being used in pollination, which is important especially in the case of wild blueberry pollination, since honeybees do not often collect substantial amounts of nectar from blueberry blooms to maintain optimal colony strength. All of these measures directly relate back to the strength of the colony and so are used to create these guidelines for hive strength standards. The Canadian Association of Professional Apiculturists, or CAPA, produces a document entitled A Guide to Managing Bees for Crop Pollination. In this publication, there is guidance on a colony standard for pollination. It suggests that a colony should have a minimum of 8 deep frames fully covered in adult bees and 5 to 6 frames of brood in all stages of development. They go on to clarify that 50% of the frames should be covered by brood and all should be housed in 2 standard deep Langstroth hive bodies. It must be stated that this is a generic guideline, not specific to wild blueberries, recommended for the pollination of orchards, but nonetheless still insightful. The Nova Scotia Beekeepers Association, or NSBA, has a fact sheet available outlining a standard for honeybee colonies used for pollination. Available on the NSBA website is a clear, concise statement suggesting 4 frames of brood, 8 frames of bees, 2 frames of honey, and 1 laying queen. Although not stated as specific to wild blueberries, this is endorsed by the Wild Blueberry Producers Association of Nova Scotia as a minimum standard. For recommendations specific to blueberry pollination, there are additional standards available. The New Brunswick Department of Agriculture, Aquaculture, and Fisheries has an excellent fact sheet which provides guidance on a standard pollination unit entitled Evaluating the Strength of Honeybee Hives. Although this fact sheet contains information related to assessing hive strength, there are also guidelines which create a standard for pollination. Generally, it states that a standard hive should contain at least 2 boxes, a laying queen, brood, and 25,000 to 30,000 honeybees. Further along in the document, there are additional details. It is stated that honeybee colonies used for blueberry pollination should have 6 to 10 frames with a variety of brood coverage present. The University of Maine publishes a comprehensive guide called Honeybees and Blueberry Pollination. This document states that a colony for pollinating blueberries should be housed in at least a 2-story hive, preferably 2 deep hive bodies, containing at least 30,000 bees, and have 6 to 10 full frames of brood in all stages of development. Methods of assessing hive strength. Experienced beekeepers can get a good impression of the strength of a colony through their own methods, but there are specific methods used to quantify specific aspects of hive strength for the purpose of comparison against a pollination standard for hive strength. These methods include various ways of quantifying frames of bees and also quantifying brood cover. Frames of bees can be measured 3 ways: by visually inspecting the bee coverage on each frame, by performing a seam count, or by counting the number of flying bees returning to the hive during a set time frame. Quantifying frames of bees provides an estimate of the population of the colony, which can be used to determine whether the criteria for number of bees is satisfied according to the hive strength standard. Brood cover is quantified through brood assessments. This is important since brood production is what determines the proportion of workers that contribute to the pollination workforce, and also because the presence of uncapped brood stimulates foraging worker bees to collect pollen and thereby provide pollination services. There are various methods that can be performed to quantify brood in a hive, including counting square inches of total brood, quantifying percent brood cover of all brood on frames, or using these methods to yield specific quantifications of brood at each development stage. These methods of assessing brood in a hive helps determine whether the criterion for brood is satisfied according to the hive strength standard. In a past ATTTA research study, results showed that hives sent to pollination meeting the pollination standard grew well and did consistently better than weak hives, showing that there are benefits to both the blueberry grower and the beekeeper to send hives into blueberry fields that meet the pollination hive standard for wild blueberry pollination. The standard of hive strength is also important as it is a contributing factor in determining how many hives should be used per acre of crop that requires pollination, also known as the stocking rate or stocking density. Present and future ATTTA research aims to provide beekeepers and blueberry growers with more regionally specific guidelines for the pollination of wild blueberries by managed honeybee colonies. This works to ensure both beekeepers and blueberry growers are hitting the sweet spot between the cost of managing and renting honeybee colonies for blueberry pollination and the benefit of increasing the abundance of insect pollinators by means of managed honeybee colonies, in wild blueberry fields.
That's great, thanks. So the focus of our next stop is going to be winter wheat, where we're going to find Caitlin, our field crop specialist.
Hi, I'm Congdon, field crop specialist at Perennia. It is June 18th, 2021, and I am standing in a winter wheat field in Kings County in the Annapolis Valley. So in this field we have a winter wheat seeding rate and planting date field scale trial, which is part of the Atlantic Grains Council on-farm agronomy program. The on-farm agronomy trials are funded by the federal government and the maritime provincial governments through CAP. Winter wheat planting date and seeding rate are factors that should be considered together in order to maximize your yield potential. A crop that's planted earlier on in that fall planting period will have more time to develop fall tillers before the winter sets in. In comparison, a crop that's planted on that later end, so something that's maybe going in after soybeans or a late planted vegetable crop, won't have as much time to develop those fall tillers. So you're going to get something that maybe only has a main stem, maybe 1 or 2 tillers before winter. That later planted crop is going to have to put on a lot more spring tillers in order to match the yield potential of the earlier planted crop, which comes with its own set of problems. Spring tillers tend to have shorter stems and smaller heads, so your yield potential right there is diminished. It's also going to have some variable development, so you're going to end up with variable maturity and moisture come harvest time. So we have 3 treatments in this field. The first was planted in mid to late September, and that was planted at 1.7 million seeds per acre. The other 2 treatments were planted in the mid-October to later October range, so right at the end of where you want to be planting in the fall. One of those treatments was planted at 1.7 million seeds per acre, same as the early planted treatment, and the other was planted around 2.1 million seeds per acre.
Back in the spring, we flew a drone over the field to see if we could see any differences between treatments from above.
2, 1. Obstacle detected.
So in this imagery, you can see a much lighter strip running through the center of the field. That is the later planting date with the lower seeding rate, noticeably different from everything else in the field. Directly adjacent to that much lighter strip is a slightly lighter strip. You can tell the difference between that and the rest of the field if you look hard, but it is much more similar. So that slightly lighter strip would be the late planting date at the higher seeding rate. So you can see how it really corresponds to the early planting date with the lower seeding rate. At this point in the season, we collect disease rating data to determine if there is a significant difference in the amount of disease present between treatments. Conditions this spring were conducive to disease development, and we found evidence of powdery mildew in all 3 treatments at this location.
Today is July 14th, 2021, and we are back in the winter wheat field to do some pre-harvest ratings. So today we're going to count the number of heads in a meter length of row, and then we're going to multiply that by the number of rows per meter width, and that's going to give us the number of heads per meter squared. After that, we're going to take 10 heads per treatment and count the number of kernels per head. When it comes time to harvest, we'll record the yield for a known area, and we'll use that number to extrapolate and give us a value in tons per acre. We'll also take a grain sample at harvest, and we'll use that to determine moisture, and then it'll be sent on to the lab for further analysis. The data from this trial will be analyzed alongside similar trials across the maritime provinces. This is the second year that we've had this particular trial in the ground, and when we plant this fall, we're going to be adding an additional treatment. So we'll have an early, mid, and late planting date, and all of those will be at 1.7 million seeds per acre. And then we'll have the late planting date at 2.1 million seeds per acre as well. And that's just going to allow us to hone in more on what's the sweet spot for that seeding rate at that planting date.
I expect the harvest data to show that the late planted at the higher seeding rate and the earlier planted at the regular seeding rate have comparable yields, while the late planted at the regular seeding rate has a lower yield.
Stay tuned to the CropLinks newsletter, as well as the Perennia and Atlantic Grains Council websites for more updates on this and other on-farm agronomy trials. I want to give a big thanks to Clay Ansems and Tim Ansems, who planted the 2 sites of this winter wheat trial here in the valley.
Awesome, thanks Caitlin. So our next stop is going to be on the topic of Christmas trees. So we're going to find specialist Jay Woodworth to see what she has to show us.
Hi there, my name is Andrew Schofield and I work at the Biology Department in Dalhousie University, and I also work with the Christmas Tree Council of Nova Scotia and with the Smart Christmas Tree Cooperative to coordinate their tree improvement program. So this program has 2 main objectives. The first objective is to find great trees. So for decades now, CTC&S has been working with growers and with various organizations to identify trees that have superior traits. So for example, we're interested in things like overall shape, color, needle retention, brush quality, spring flush time, and also how well they hold their needles after they've been harvested. We're preserving these great trees by grafting them to various orchards around the province. The second aim of the tree improvement program is to make varieties of balsam fir trees. So for example, this is a really common practice in agriculture. So many consumers would know about apple varieties like Honeycrisp, Gala, or Ambrosia. And perhaps more importantly, farmers know the performance of and the particular production requirements of these various varieties. We create these balsam fir varieties using a tissue culture technique called somatic embryogenesis, or SE for short. And although it sounds pretty fancy, it's actually a very old technique. So we're not altering the genetics of these trees or creating GMOs. All we're doing is taking the young, developing seedling from out of the seed and causing it to make multiple copies of itself. The result is hundreds of thousands of natural trees. And since they're all genetically identical copies of that original tree, they constitute a new variety of balsam fir.
Good morning, this is Jay Woodworth, the Christmas tree specialist with Perennia. I am here today standing in one of our CLMT research sites. So this is one of 9 field evaluation sites for the Smart Tree Development Program. So we had funding to set these sites up through the Canadian Agricultural Partnership's Crop and Livestock Management Trials. We were able to put 3 in each Christmas tree region across the province. So we've got 3 up in Lunenburg County area, we have 3 here in Cobequid area, and we have 3 up in the northeastern region, which covers Antigonish and Guysborough counties. So you can see here each of these trees. So this one's about 3 foot in height. There's one on my left hand side as well that's a little bit taller. What we're looking for in the Smart Tree program is trees that are late flushing. So we'll pan in a little bit later on this tree to my left, but you can see it's the middle of July and this tree is still pretty fresh, pretty new with the flush. So that's great news. We also look at needle length, so we like to see trees that have longer needle length. We also look for needle retention, so there's a fine balance in there, getting trees that express both characteristics. We look at color, so often we go for either that really deep dark emerald green color or sometimes the blue tinged or silver tinged under needles. So the trees that we're finding through the Smart Tree Development Program, we have identified crosses, so parent trees that have really good characteristics from Nova Scotia mainly and from New Brunswick in some cases. We've strategically crossed them so that they're expressing genetic traits. So it's an entirely natural process. It's just a little bit different because we're actually growing up these seeds in a petri dish and getting them ready for mass production. So the trees that are here, this is the first time that we've been doing a field evaluation on the trees. So these were grown in the lab and now they're planted out in a natural site to see how well they do in a natural environment that we would see across the province. So we're going to walk around and show you some of the different lines. Keep in mind we are just in Cobequid, so the different lines will express themselves differently depending on what environment they're planted into. So if you're looking to plant smart trees in Lunenburg County, for example, it would be more useful to look at the Lunenburg data, which is maybe where we should have gone today because it's raining in Truro again. Barcode tag here, and we bring the barcode scanner out with us when we go to evaluate the trees. So we're just able to scan the tree identification directly into our spreadsheet, and then we evaluate them for some of the characteristics that I discussed earlier. This is tree 242. So this is one of the older crosses in the Smart Tree program. It's expressing some really good budding. So you can see if you go through and count the internodal budding, we've got 1, 2, 3, 4, 5, 6, 7 going right on. And for a tree of this height, that's showing some really good budding. We're also starting to see some nice color. This tree was a late flusher as well for Cobequid region. So that's a good characteristic to keep in mind. And it's staying relatively healthy for being planted out in a site like this. We did have some trees that were hit with frost in this area. Some of our blanks are looking pretty hard, but these ones look really good. So we're happy to see that some of the smart genetics are successfully being expressed in the field. It's going to be a 10-year cycle to get this tree to fully harvestable rate, but we're still really happy to see that even in just a couple of years of field evaluation, we can see some of the characteristics that this tree was bred for. Alright, just going to show you some characteristics on this tree here. So this tree, quite a bit bigger, hasn't been sheared yet this year. It could really use it. You can see how much growth we had this summer here in Nova Scotia. Just really phenomenal. What I want to point out on this tree, it's a lot easier to see some of the characteristics that we actually evaluate our tree genetics for when they're a little bit larger. So this tree, very uniform. Pretty hard to see the stem here, so there's a lot of stem coverage, which means we've got great density of foliage throughout this tree. You can also see there is quite a bit of bottled brush happening on this guy, so you've got your needles 360 degrees around. And on some of this newer growth, you can see when I hold this branch up here, there's a little bit of a silver or blue flash on the underside of the needles. That's really desirable with consumers, so that just gives you a little something extra special with the tree. So to have a balsam fir that is expressing really good needle retention, is showing really good foliage characteristics, and also has that nice silver blue flash, that's really great for the industry here in Nova Scotia. So just wanted to take a minute and point that out on some of the larger trees that we have in the program. This tree here is another smart tree line, so this one would have been a little bit younger. It's been planted out more recently. This is one of the replicates of 238, so there aren't a lot of 238s planted around the province. So we're just getting into the field evaluations with these guys. This hasn't been sheared yet, obviously. We're seeing some pretty good growth, pretty uniform tree, nice bottle brush that you can see here. We're still waiting to see what the needle length is going to be, but overall, healthy tree. And if you look around, some of the other conventional seedlings that are here have suffered a little bit more insect and disease damage, so it's nice to see that the smart trees are still doing well. So here's another 238. This one would have been a little bit older than the last one that we had just looked at here. So it's when they start to get older, it's a lot easier to see some of their phenotypic characteristics. So you can see here this tree's got pretty good bottle brush, quite a bit of internodal budding. It's going to have decent needle length, and the uniformity of this tree is just pretty good as well. This one could use a shear, but other than that, just looking at the buds that are forming on that tree is really nice to see.
Well, thanks so much for joining us today on the topic of optimizing production for the tour. And we still have one more tour left, and it'll be with Talia in bus number 2 next week. So hopefully you can join us then. It'll be on livestock management.